Hi everybody, I'm Chiara La Tessa.
I'm Gianluca Lattanzi.
We are from the Department of Physics at the University of Trento in Italy, and we are here today to present you the study plan of biological and medical physics. In this presentation, we'll give you an overview of the research activity related to this area, which are carried out at the department, and then we'll give you a list of courses that we recommend you to attend if you're interested in following this path. And most importantly, at the end of this presentation, I'm sure that it will be convince you that of all the possible places,
You should come here.
Hi everybody, and welcome to the presentation of the research activities in the biological and medical physics area in our department. I will first describe the research activities in biological physics. For each activity, you will see the name of the principal investigator, or PI. Please refer to this name if you want to receive further information on a specific research activity. First, let me introduce Professor Albrecht Haase. Albrecht is working on biophotonics and neurophysics and is employing experimental methods, and in particular multiphoton imaging, to study odor perception in insect models. You see here some beautiful images obtained by Professor Haase laboratory of the honeybee antennal lobe, for instance, here on the left, and also of the identity information, what is called the odor identity information across several subjects in insects again. Albert is also very active in trying to apply quantum biological models on, for instance, the vibration theory of olfaction. Here you see a specific example of this technique, and is also studying and trying to characterize magnetoreception in some specific neuronal cells. In addition, Albert is also employing optogenetics to induce, by using laser light, artificial stimuli and artificial memory in neurons, and in this way is also able to investigate neural network plasticity during sleep. Professor Marina Scarpa is working on several applications of experimental techniques to the research area of nanomaterials. Here you see two pictures obtained by Professor Scarpa, and in particular here you see on the right sponge-like microparticles of porous silicon. And these particles find several applications in drug delivery and bioimaging. But Professor Scarpa is also employing these techniques to study hydrogels, and these hydrogels are obtained by self-assembly of bio-nanostructures. You see here an example on the bottom left of this slide, and these are patches that can be used as therapeutic dermal patches for in applications, in medical applications. And then we have several researchers that are active in the theoretical physics group. For instance, here you see the activities by Professor Pietro Faccioli. Pietro is a theoretical physicist and is trying to apply several methods and several theoretical physics methods to different problems of biological interest, but not only. For instance, he's trying to apply quantum computing for soft and biological matter, but is also an expert in statistical mechanics for big data analysis, for instance, clustering or stochastic modeling, with several applications in biophysics. He's also developing advanced algorithms for molecular simulations, and is applying quantum field theory to open systems like biological materials, but not only. For instance, there are applications in ultra-cold systems or molecular systems. Luca Tubiana is active in the characterization of several soft matter systems, here used in particular polymer materials. Luca is trying to characterize the properties or the topological properties of soft and biomaterials. You see some of the examples here. For instance, these materials are very interesting. They are called Olympic gels because you see that they are formed by several rings that resemble the Olympic rings. Raffaello Potestio is the principal investigator of a research project that has been funded by the European Research Council. This project is called Variables, and the aim of Variables and the aim of Raffaello's research is to employ modeling, coarse graining strategies, and information theory to biophysics and to problems of biological interest, like, for instance, protein structures, protein dynamics, and also protein assemblies. Finally, myself, I'm using molecular dynamics simulation to characterize some biological materials, like, for instance, membrane proteins that are represented in these pictures here, but also organic materials like polymers that are employed in organic photovoltaics. And with that, now I leave the stage to Chiara.
Hello again, everybody. Now I'm going to illustrate to you the research activity that myself and my colleagues carry out at the Biomedical Radiation Physics Lab at the Department of Physics, also known as BEAMER. The central focus of our research is the investigation of the interaction of radiation with matters and the effect that this interaction has on biological systems. The first step of this chain of events are the electromagnetic and nuclear interaction of radiation with matters. The product of this interaction are the one that trigger the mechanisms on the biological tissue and eventually lead to the biological effects. We study both the interaction and the effects on different scales. So from the nanometric scale, which is the size of the DNA structure, up to a macroscopic scale, which is our entire body. To investigate these topics, we use different tools, which include both experimental, computational, and theoretical weapons. The interaction of the radiation with matter are characterized with detectors, coupled with advanced electronics to read out the detector outputs and to perform the data analysis. Furthermore, to obtain a more sophisticated analysis of the results and analysis of the data collected, we use machine learning techniques. In addition, we also use different Monte Carlo transport codes to help in understanding the data analysis and to fill the gap of the experimental data. We then put everything together by developing theoretical models to predict the interaction radiation with matters and the biological outcome for different irradiation scenarios. There are two main applications to our field and to our research. One is the radiotherapy for cancer treatment. We study different aspects of this in this field, considering both neutral radiation like X-ray and charged particle radiation. Another application is radioprotection of astronauts in space, which is one of the right now is one of the showstopper for space exploration and, for example, for a possible mission to Mars. The research projects carried out by BEAMER are divided into three groups. One led by me, one led by Dr. Emanuele Scifoni, and one by Professor Francesco Damasino. I'm going to give you an overview of the project that I lead
and then pass the microphone to my colleagues. One of the central points of all the research activities of my group is the characterization of the radiation field, where characterization means the quality of the radiation and the quantity. So the amount of radiation that the specific target, whether the target is biological or non-biological, receives. To do this characterization, we consider two approaches: dosimetry and microdosimetry. The difference between these two approaches lies in the volume where the characterization is done. In microdosimetry, the volume of interest is extremely small in the micron scale and simulates the volume, for example, of a cell nucleus where the DNA is contained. Dosimetry instead, the volume is much bigger and could be, for example, the entire cell population, an organ, or even the entire human body. The characterization of the radiation field with microdosimetry or dosimetry can be coupled with radiobiological measurement, which assess and characterize the effect of this of radiation on biological system, as for example, a specific cell line or even in vivo measurement. And the combination of these two is the input for developing models that describe the biological effectiveness of radiation and can predict the risk associated to exposure. This workflow can be used to for the different application, both in radiotherapy or in radioprotection for space exploration, and currently is the structure of is related to two projects. A microdosimetry-based assessment of biological effectiveness in ion therapy, and CASPER, whose team is shown in this picture. Both microbit and CASPER are focused on radiotherapy, and their outcome can help verify and optimize treatment plans in ion therapy. Another application specifically related to radiotherapy consists in developing a method to verify the treatment plan online. So while the treatment plan is actually delivered, with the possibility of correcting it in case of misalignment of the patient. In our project, we want to achieve this verification by exploiting the interaction of the radiation with the human body and the production of secondary particles. In particular, the particle that we are considering, the radiation we're considering, are gammas, whose production we're going to enhance with a specific element that is going to be selectively carried inside the tumor. In this project, we developed an experimental system to actually monitor the production of this gamma and to correlate the production of this gamma with the position of the tumor. We also use Monte Carlo simulation and modeling to couple it with the experimental data and to develop a new methodology which could be applied in the clinics. The last topic my project and the project of my group are focusing on is the optimization of shielding materials for space exploration and for different types of space missions. So ranging from mission on the ISS, so close to Earth, to longer mission to the moon, and even to interplanetary mission to reach Mars. The most common approach for designing a radiation shield is to minimize the radiation dose to which the astronauts are exposed to. So the lower is the dose, the best is the shielding effectiveness in protecting the astronauts. We propose a new methodology where, together with selecting material for their shielding effectiveness in terms of dose decrease, we also consider the best material dose that at the same dose decrease yield lower side effect or yield a lower risk of developing side effects related to radiation exposure. Of course, the choice of the shielding material depends on the radiation field, and therefore we consider different optimization depending on the mission scenarios and on the mission duration. With this, I am done with the overview of the project I lead, and now I leave the floor to my colleagues.
Hello, everybody. I am Emanuele Scifoni, and I'll present a snapshot of my activities within the BIMER group and in collaboration with several international partners. So my major interest of research is the mechanistic understanding of radiation physical action. And this you can see here in this big plot where you see all the processes which follows the primary ionization from the radiation, follows by the excitation and the particles generation, radical species, including physics and chemistry. This is my major focus, where we want to understand how specific mechanisms which are related to some process related to biological damage can be explained. Here you see, for example, some cartoons of how the temporal evolution of the damage plays. So this can be applied to several types of radiotherapies. For example, the possible increase of radiation action with nanoparticles, called nanoparticles, which generates an increase of electron emission, and this can give several steps, several pathways of radiation enforcement and better improvement than of the therapies. The other focus of my research is the biological treatment planning, which means the use of different and novel approaches based on ion beams, in this case, for example, multiple ion beams, to optimize the treatment and to exploit better the major features of biological and physical peculiarities of different beams in order to get the better effectiveness on a tumor of a patient. Thank you for your attention, and I wish you a wonderful master thesis.
Hi, my name is Francesco Tommasino, and I am part of the BIMER group, Biomedical Radiation Physics. Here I just want to spend a couple of minutes to describe some of the research activities on which I am currently involved. One of that is the FOOT experiment, financed by INFN, the National Institute for Nuclear Physics. This is an experiment, nuclear physics experiment, dedicated to measure nuclear interaction cross-section that can be applied to improve the current description of this process in the treatment planning software, what is finally used to calculate the treatment plan in radiotherapy with protons and with charged particles. So we expect that from this interesting applied nuclear physics experiment, we can learn something that can improve the quality of proton therapy treatment. Similar to that, but from a different perspective, is the XP Calibre experiment, also financed by INFN. This is an experiment that we are performing in collaboration with the INFN Firenze. And this is dedicated to trying to translate into clinical activity this system for proton CT, so the computed tomography that currently is performed with X-rays, here is proposed to be performed by means of protons. And this is due to a number of reasons that finally are linked to the fact that by using proton CT, you will be able to better describe the stopping power of human tissues in a treatment planning. And also here, final goal will be to improve, to enhance the accuracy of proton therapy treatments. So you see that here we are taking a different topic, imaging performed with protons instead than X-rays, again with the final goal to reduce the uncertainty in the therapy treatment and to improve the quality. Last but not least, during these years, I have performed several studies in collaboration with the medical physics staff of the Trento Proton Therapy Center, especially studies dedicated to treatment planning application. For example, we investigated how we could improve and extend the quality of proton therapy treatment, for example, to specific targets, for example, for breast cancer. And from this point of view, we performed a number of studies, pure treatment planning studies dedicated to find the optimal irradiation technique, dosimetric studies trying to find out which could be the main uncertainties in this type of treatment, modeling studies, for example, studying the risk of secondary cancer that are related to breast proton therapy treatment. This is just to mention a few of the activities in which I'm involved. And as you can see, they span from experimental activities to modeling and medical physics related ones. I hope you found that interesting enough.
Finally, here we provide a list of courses that you can choose to attend this study plan in biological and medical physics. First of all, there are two mandatory exams, mandatory courses, one in experimental methods and the other in quantum mechanics, fields and symmetries. Then you have to choose 5 courses among those listed here. You see that there are some rules. You have to choose 1 course in the area called Phys02, which stands for theoretical physics, 3 courses in the area either Phys03, which is physics of matter, or Phys04, which is nuclear physics, and then 1 course among this list without, for this course, you are free to choose any course that we recommend within this list. So basically what you have to do is to choose 5 courses out of these 8 here in this list. Then you have to choose again 4 courses in this list that we provide to you here. You see that there are 11 courses you can choose from, and you can take 4 of them, but you also have 2 courses at your free choice. And this is something that, so either you can take these exams from these courses from the above list, this one, or even the other one here, or you can choose any course in the University of Trento. And of course, for any information, please refer to Chiara and to myself. Thank you.